Charlie, and I'm delighted to be here. You just have to excuse me, I should be asleep at the moment. So today what I'm going to talk to you about is GABA, and I'll have a bit of an excursion into glutamate as well, because we all sort of think we understand what glutamate's doing, so we'll sort of skate over the top of that and then talk about excitation and inhibition, because glutamate is the major excitatory neurotransmitter in the brain, and as we know, GABA is the major inhibitory one, so you have to think about the two of them together. Then I'll talk about the energy budget of the brain, so how much glucose and oxygen it has to burn in order to do its various tasks, which of course is an estimation because it's continually being revised every time people do new experiments, but it's relevant here. So we'll have glutamate and the bold response, and then we'll talk about inhibition and the energy budget. So how expensive is inhibition compared to excitation? And then there'll be a bit of neurochemistry, and then we'll talk about the power of GABA, because it is actually a very powerful molecule. So glutamate and GABA, are they the yin and the yang of brain metabolism? So they're very similar molecules. You've seen the structures before. So they're connected just by a single enzymic reaction, a glutamate decarboxylase, which basically takes that little red molecule that you can see, the carboxylic acid, off. It's decarboxylated, you lose it as carbon dioxide, and you end up with GABA. So I have a poll now, so be interesting to see what happens. The question for you, it says this slide will display live content. Okay, well the question for you is actually, what is the proportion of excitatory to inhibitory synapses? And this should be loading the poll, and it isn't. So the question for you, which I'll just see if I can remember what the options were. There was one, is it 50-50? Is it 90-10? Is it 97-3? Oh dear. Is it 60-40? Any idea? Who votes for 50-50? Who votes for 90-10? 90% excitatory, 10% inhibitory? Who votes for 97-3? Who doesn't care? Okay, let's see if I can move on. The answer, which would have come up if everyone had voted, is actually 90-10%. So about 90% of the synapses in the brain are excitatory and about 10% are inhibitory. So if we look at the brain energy budget, so this is taken from David Atwell's work, and David's very good because every now and then he does these little calculations about what the total brain energy budget is. So if you're using up a certain amount of oxygen and glucose every day, what proportion of it is used for housekeeping reactions, what proportion of it is used for synapses, how much does the grey matter use compared to the white matter. And he's basically come up with this idea that the resting potentials and housekeeping using up a certain amount of the budget. So in white matter, it's a lot more as a percentage because the white matter isn't doing as much in terms of synaptic activity. In the grey matter, which is the one here on the left, he's got the housekeeping activities, which is things like lipid synthesis and moving your actin around and reaching out to make a synapse with somebody, as taking up about 25% of total glucose use. Resting potentials and action potentials add around, that's these APs and RPs, and the synaptic activity at about 43%. So that's all predicated on it mostly being glutamatergic activity. So it's quite expensive to run. It has been known for some time that there is coupling of brain glucose metabolism and glutamatergic neuronal activity. So Nikki Simpson showed this quite nicely in a paper from some time ago, in 1998. And this has been repeatedly shown by various people that there is this relationship between glutamate and glucose use and glutamatergic neuronal activity. So more excitation means more activity. Duh. Okay. The glutamate levels and the bold response tend to couple quite nicely. You can see here you've got an on-off stimulation, which fMRI people are familiar with, and your glutamate signal tends to track your bold response actually quite nicely. It is to a degree area dependent, and it is to a degree dependent on what your stimulus is. So you need to bear those things in mind. Of course, you know, in neurochemistry, nothing's simple. There's always complications. It could be this, it could be that. So you have to take these things into account. But generally, the two things correlate quite nicely with one another. So, you know, you'd actually expect that because this is the neurochemistry. We've got our Krebs cycle here. So isocitrate coming around to alpha-ketoglutarate and going off to bisuccinyl-CoA. So you can just imagine the Krebs cycle sweeping around here. And glutamate is in direct exchange, really, with alpha-ketoglutarate, which is one of the main control points in the Krebs cycle. It's also, the other thing that we often forget is these aminotransferase reactions, which convert glutamate to alpha-ketoglutarate, and in the process we'll add an amine onto things like oxaloacetate or onto pyruvate, and you end up with various amino acids like aspartate, which is also an excitatory molecule, and alanine, for example. It also directly makes glutamine, but these two reactions are cell-specific, and the synthetase actually requires ATP. So you're not in glutamine and glutamate are not necessarily directly connected to one another, whereas glutamate and alpha-ketoglutarate do tend to be, because these reactions here, particularly the aminotransferase ones, are fairly near equilibrium enzymes. So this means that if your TCA cycles, by the way, don't necessarily go faster, they tend to get fatter. So it's like a donut getting fatter. You put more molecules into the TCA cycle, which is how you get more flux through it. It does speed up, but it also gets fatter. But as it gets fatter, your alpha-ketoglutarate concentration increases and your glutamate concentration increases concomitantly because these reactions are in fairly fast exchange. So the bottom line for that is that glutamate and the TCA cycle are fairly nicely linked, and therefore brain energy metabolism and glutamate are fairly nicely linked. But what about GABA? People obviously think that GABA is important. If you look at the commercial activity around GABA at the moment, there's lots and lots of people busy flogging it off. This idea that you could eat it and it would end up in your brain seems a bit strange because it doesn't actually cross the blood-brain barrier. So if you're ever interested in buying these things, just remember that it's probably all just going down the toilet, not actually into your brain in the first place. But it's a big market. So what is GABA actually doing? So we've got a little bit of useful background facts here. Like I said, there's about 90% of cells and synapses are glutamatergic. So the other thing you want to consider around energy budgets is that postsynaptically, chloride ions, which is what you move when you have an inhibitory synapse, they move down a smaller electrochemical gradient at inhibitory synapses than sodium ions do at excitatory synapses, which tells you that there's probably less energy expenditure involved in hyperpolarizing as compared to polarizing a membrane. So there's various different types of inhibition, and this is important when you're considering what the GABA is actually doing. So you have presynaptic inhibition, which is what happens at the presynaptic terminus of a synapse. You also have postsynaptic inhibition, which is at the postsynaptic terminus. You have phasic inhibition, which is basically the sort of normal spiking GABA that is very short-lived. And you have tonic inhibition. And tonic inhibition is where you get sort of tone that goes on for some time and helps to control your entire network. So I'm going to talk a bit about the various types of inhibition because they're important when you're considering what your GABA is actually doing. Now I have another slide which probably isn't going to work. So the question was, what is the estimated relative energy cost of inhibitory synaptic activity? So we're talking now probably mostly about the phasic inhibition. They're the options, 10, 16, 3 and 0.5%. So remember that we said that the in grey matter housekeeping reactions were taking up about 25% of activity. How much do you think inhibitory synaptic activity is actually contributing? Well, this is good because the answer is actually 3%. And eventually we'll move on to the next slide. Thank you. So 3%. If it's this small, why do we need to bother about it? If it only accounts for 3% of brain energy, then as scientists, what's it matter? Well, because 3% of the brain activity is the synaptic activity, but there are a number of ways that GABA can actually significantly impact what's going on in the brain, and the rest of my talk really is going to be taken up with telling you about those. So the answer is yes. Well, I would say that, wouldn't I? So okay, the devil is in the details. We have a couple of metabolic pathways here, which we need to have a look at. So what we have here is the presynaptic neuron that I mentioned, the postsynaptic neuron, and an astrocyte. There's also oligodendrocytes in the brain, which actually are now emerging to have quite strong metabolic activity, but all the models actually ignore them. So just be aware of that. So GABA, as I said, is produced in the presynaptic neuron, for example. It has to be a GABAergic neuron because if it's not a GABAergic neuron, it doesn't have the enzyme glutamate decarboxylase that actually makes GABA. So it's made in GABAergic neurons. It's produced from glutamate, and it can be incorporated into a vesicle where it can be released at a synapse as GABA. It can then has a number of different fates. It could go and bind to a particular type of GABA receptors, the GABA A receptor. Now, the GABA A receptor, when I say the GABA A receptor, there's actually multiple different types of GABA A receptors. It's made up of alpha, beta, gamma, delta, epsilon, pi, and rho subunits, which there's multiple different versions of each of them, like there's 6 alpha subunits, for example. Luckily, you have to have an alpha, a beta, and a gamma subunit, and then you could possibly include one of the others. And that sort of narrows down the number of possible GABA A receptors we could have to around 10,000 different types. In practice, there's probably only about 13 to 15 different types of GABA A receptors expressed in significant quantities in the brain. But just be aware that all GABA A receptors are not equal and they all do different things. So you have GABA A receptors which live in the postsynaptic neuron, but you also have GABA A receptors which live out here, which is what we call perisynaptic or extrasynaptic receptors, and they play very interesting and specific roles in inhibition. We also have the GABA B receptor, which luckily only has 2 subunits. There's the B1 and B2. Sounds like the bananas in pyjamas, but and then you have a couple of different splice variants of those. And those are metabotropic receptors. So instead of letting ions in and out, they're coupled to a G protein, which then affects the local environment, so calcium channels or chloride channels. The other thing we have here is these GATs, which are Gaba Transporters. So they're responsible for removing the GABA from the extrasynaptic space or from the extracellular space once it's been released. So if you have release of neurotransmitter, then your transporters will actively take up GABA. And the interesting thing about these transporters is that they can actually work in reverse. So they can release GABA. And they do that depending on what the local chloride potential is. So they're activity dependent. So if you've got activity, you can have release of GABA. So just bearing all those things in mind, we also have a way of metabolizing GABA once it's been made through Gaba transaminase, which is an enzyme that basically reincorporates GABA back into the TCA cycle at the succinate level. And that'll become interesting in a minute as well. The other, now this looks awful, but I'm not going to go through this. Phew, everyone says. Astrocytes can actually also make GABA. They don't make much, but you need to be aware of it. So it's made through this cute little pathway over here, putrescine, which sounds horrible. It's actually a type of biological amine, and it's active at NMDA receptors. But ornithine, if you add ornithine to brains, you actually end up with quite a lot more glutamate and GABA, either through this pathway, which is the main one, or through the metabolism of putrescine, which is the bit that can occur in astrocytes. And that links GABA metabolism through to creatine metabolism, arginine, and nitric oxide. So that's another little pathway that people just usually ignore because it's complicated and it's not really known what activity, what the impact of it is. So back to this pre- and post-synaptic activity at GABA B receptors. So the presynaptic activity can change the amount of calcium that you've got. And the calcium is important for basically release of neurotransmitter. So if you have GABA binding to the GABA B receptor, it changes the calcium levels and basically means that the presynaptic neuron releases less neurotransmitter. So this is a way that GABA can interact with the glutamatergic system or with the release of neurotransmitter. So these GABA B receptors could also be on non-GABAergic neurons. The postsynaptic one is linked to potassium channels, and when it activates the potassium channel, it means that the action potential that you get through the postsynaptic neuron is actually can be lowered. So it has a way of sort of turning off specifically the postsynaptic neuron. So what does this actually mean in terms of the energy budget? So this is the calcium channel, and this is the chloride channel, and it has a negative effect. So in terms of the energy budget, so this is a hypothetical. So if we say resting potential, 80% of our activity is excitatory and 20 is inhibitory. We actually know it's more like 90-10, but for the sake of this, we'll just pretend it's 80-20 because the numbers are easier to deal with. So if we apply an agonist at GABA B, if the agonist has only a slight effect, what it tends to do is basically decrease the excitatory but increase the inhibitory response. Now, inhibition is not an energy neutral or an energy negative thing, because to inhibit a neuron, you have to hyperpolarize it. And that means you have to be pumping sodium and potassium. So there is an energy cost associated with it. The resting potential is it's called resting potential for a good reason, because it's the most energy efficient place for the neuron to be. So if you hyperpolarize, you have to pump ions, and that takes a bit of energy. So in this case, we've turned a bit of excitatory down, but we've got more activity at the inhibitory neuron because of our hyperpolarizing. And in fact, that lends to a slight increase in the overall energy budget. So we've got basically an increase with the agonist. If we turn the inhibition up slightly more, we get a bigger reduction in excitatory activity for pretty much the same energy budget, and you get an overall decrease in metabolism. So an agonist at GABA B could give you an increase in energy demand, or it could give you a decrease in energy demand. Ditto with an antagonist. At this point, everyone has to stand on their head to understand what an antagonist is doing. So it's actually switching off the off switch. So your agonist is actually basically increasing your excitation with a decrease in inhibitory activity. And depending on how much you put in, it also can give you more or less energy demand. So an agonist or activity at GABA A receptors, just because you've turned the receptor on, doesn't necessarily mean you've got less total energy demand. It's a bit complicated, but that's how it works. So what about tonic inhibition? Well, that's the really interesting thing for us using MRS to measure GABA. So with tonic inhibition, what we're talking about here is extrasynaptic release of GABA by these transporters, and there's also there's various bits of evidence in the literature supporting extrasynaptic release of GABA. So once it's released, it can bind to either these extrasynaptic GABA A receptors, and they tend to be more of a master switch. And to show you about that, I'll show you some results from an experiment that we did a while ago. So we used a compound called vigabatrin, which inhibits Gaba transaminase. So it stops GABA being recycled back into the TCA cycle. And what that does is it increases extrasynaptic concentrations of GABA. So not the GABA that's sitting there in the vesicles, but the GABA that's actually hanging around being metabolic GABA in the extrasynaptic space. So put vigabatrin in and your GABA concentrations go up because it's a nasty inhibitor. It kills the enzyme, basically. So these are experiments with brain slices, and you don't need to know any of the details of this apart from up is excitatory or more metabolism, and down bars are less metabolism. So if we put AMPA, which activates the glutamate AMPA receptor, into brain slices, basically we get an awful lot of metabolism. If we put this thing in, which is acts at metabotropic glutamate receptors, we get a big increase in metabolism. If we put this in, which is a GABA B antagonist, we get a mild increase in some bits of metabolism, but not in others. If we put these drugs in along with vigabatrin, so vigabatrin's upped our GABA levels, and we've put these activators in, and we get a big decrease, and we get exactly the same big decrease with all of these. So no matter how you activate the slices, as soon as you have more GABA, once the slices are activated, you get a big turn off of everything that's going on. But the interesting experiment is this one, which is where we added in this compound called TPMPA, which is an antagonist at GABA A receptors and also at GABA C receptors, which are actually a type of GABA A receptor that has Rho subunits only. And they're very, very sensitive extrasynaptic receptors, so they respond to very low concentrations of GABA, but they're not in the synapse. So it's where you get this overspill of the GABA coming out of the synapse. So you've got too much activity, you have all this GABA being released, and it activates these receptors. And when you put TPMPA in, it has normally got a very strong excitatory response, but when you put it in with the vigabatrin, they cancel each other out, which tells us that wherever TPMPA is acting, and unfortunately, like most GABA drugs, it's not clean, it acts at more than one receptor. So we don't quite know what the culprit is, but we know it's got to be some sort of GABA receptor. We get this very nice, almost nothing happening activity. So that shows you how powerful GABA can be. So you've gone from really, really active to everything switched off, just with a tiny little bit of extra GABA. And this has been shown to be cell-specific and act through various receptors. Most people are pinging the alpha-6 containing GABA A receptor, but yet again it still hasn't been nailed down. GABA can also directly modulate blood flow, so there's quite a bit of evidence now that it directly affects the vasoconstriction. It may do this itself through GABA A receptors, or it may do this through a neuropeptide signalling system like neuropeptide Y. And I've just put a few papers up here. There's a plethora of them in the literature about how GABA can impact cerebral blood flow. So, what do bulk GABA levels actually mean? It's a good question, and one that we're going to have to keep asking, but it's brain region dependent. So depending on, remember every time you place a voxel in the brain, that voxel is going to have a composition of multiple different types of cells. So MR people see things in black and white. So we've got grey matter and we've got white matter, and we get really obsessed with parcellating our voxel. That voxel also contains multiple different types of neurons and oligodendrocytes, which people ignore, and astrocytes, and different populations of GABA-containing cells. So it is very brain region dependent what happens with GABA. It's also activity dependent. As I showed you, if you activate the slices, you can actually then have this feedback mechanism whereby the GABA is released and everything gets switched off. And I think my colleague over there will be discussing this. You cannot distinguish neurotransmitter and metabolic pools. Every time I read a paper where people say, I've measured neurotransmitter GABA, yeah, you can't. The two things are certainly not in a 27 cubic centimetre, well, centimetre cube box, which is what you're measuring with megapress. The neurotransmitter and the metabolite are in fast exchange. So it can be a neurotransmitter 1 minute and it's back to being metabolic GABA the next. So you can't distinguish between the two, and you've got no way of knowing what the proportion of each is because it's a bulk measurement. It does seem that total GABA levels are related to this tonic inhibition, and the more GABA you have, the more likely you are to be able to turn things off in a hurry. If you have low levels of GABA, the amount of GABA that you've got available to release through the GATs, through the transporters, is relatively low. But of course, the research is ongoing. We're finding out new things about the GABAergic system all the time, and every time people publish papers having measured GABA in vivo, it adds to that literature. So go and do it. Go and measure it. And I thank you for your attention. Lactate's a bit of a tart, really. It goes anywhere and everywhere, and people discuss lactate as being an anaerobic. In the brain, most lactate production is from so-called aerobic glycolysis, so it's through sort of decoupling of oxidative activity and glycolysis. But lactate dehydrogenase itself is a near equilibrium enzyme, so lactate and also lactate transport from the cell is also just a flip-flop thing. So your levels of lactate don't really tell you anything much apart from levels of lactate. In my slices, they tend, because the amount of lactate that can escape is always controlled, they tend to tell you things like what the pyruvate clearance rate is. So the amount of lactate that you get is related to how fast you're putting your pyruvate down into the TCA cycle.
Great, thank you very much.
Our next speaker will be Professor Charlie Stagg, who's a professor of human physiology at Oxford University. And she today will be talking about changes in GABA, neuroplasticity, and learning. Thank you.
Great, thank you very much. I was taking furious notes during Lindy's talk, so I now need to refocus and tell you a little bit about what we've been measuring with spectroscopy and what that might be able to tell us about learning and plasticity in vivo in the human. So the first thing then is, why on earth might we want to measure this? Why everything anybody knows from basic neurophysiology 101 is that plasticity is glutamatergic, surely. So why should we care about GABA in this context? I'll then cover a little bit of the literature about motor learning, where this has been studied most in humans. I'll go on to talk a little bit about some work around looking at learning outside the motor system, particularly perceptual learning. We'll then go on to talk a little bit about long-term plasticity and some evidence as to whether the kind of things that we're seeing in the short-term studies that tend to be what we perform are still present long-term, and there's still some significant questions around that. And then a little bit towards causality. I'm not for a second going to get there, but cover a couple of ideas that are around in the literature where people are beginning to get towards it. And hopefully with the idea that you will can take something from those studies and go away and, as Lindy says, do the next experiments and work out how much closer we can get to trying to pin down whether or not what we're measuring with GABA spectroscopy is actually telling us what we want it to tell us, and whether we can get to the point when we know whether or not GABA is causal for plasticity. Okay, so little backtrack. So why on earth would we begin to think that GABA changes might be important in plasticity? There are an absolute ton of papers that I could start off with, but I've edited it down for the interest of time. And I'm going to start off with just one. So the first thing I'm going to do is show you this, which is a slice through the cat motor cortex. And in green are the pyramidal output neurons, but in red is a stain for GAD, the enzyme that produces GABA. And so these are the Gabaergic Interneurons. And Lindy's made the point very beautifully, and I'd just like to echo that we obsess about glutamatergic neurons, but actually the GABA is a widely distributed, beautifully diverse set of interneurons that have really complex effects on glutamatergic firing. And the multiplicity of different types and the very specific synaptic patterns that they seem to produce allows us to think perhaps that GABA is a really, really specific and sensitive way by which the brain can modulate ongoing activity. I'm going to talk about some basic neurophysiology. So this is some long-term potentiation induction in a freely moving rodent. This was work done 20 years ago now, gosh. And what they did is a very standard LTP experiment. They stimulated presynaptically and recorded postsynaptically. And for a given input, if you don't do anything, you get a fairly consistent output. What they then did at the dotted line is induce long-term potentiation like plasticity in the cortex, so totanically stimulate presynaptically and postsynaptically to strengthen those synapses. And that develops a little bit in the freely moving rodent, but you can see that you get very standard LTP induction, where for a consistent input, you now get a much stronger output for the postsynaptic cells. Okay, so classic neurophysiological demonstration of synaptic plasticity in action. The interesting thing about this study is they then went on to do that exactly same experiment in the presence of a GABA agonist, or a drug that increases GABA concentration, and what they saw was absolutely no effect at all. Okay. So this doesn't mean that synaptic plasticity is a GABAergic process. It isn't. There are entire journals dedicated to NMDA receptors, to glutamate, to AMPA, to the very complex things surrounding glutamatergic plasticity. But what it might suggest is that a decrease in GABA allows an increase in cell firing and therefore acts as a gating mechanism by which glutamatergic LTP-like plasticity might occur. And that idea has spawned a number of studies in the literature trying to unpick that and see how far we can get to use that idea to look at different aspects of plasticity. So this all started off really in the motor learning field. There are a number of reasons for that, but mainly that it's really easy to do learning of motor skills. Most of this work is done at 7 Tesla now, and we've heard about the reasons for that. Those I'll be dipping back and forth between different scanners, different sites, different field strengths. So there was a study, about 15 years ago now, from Oxford, before my time, where they performed a motor learning task and showed a decrease in GABA during motor learning. For various reasons, that was the first demonstration of dynamic changes in GABA, but for various reasons it had been quite difficult to replicate. So very recently, James Kolosinski, who's now down in Cardiff, and my group tried to look at the question of if GABA decreases are important to allow plasticity to occur, if we do something that induces plasticity, and in this case this was a serial reaction time task, it's a motor learning task. We asked our subjects to learn a 16-button press sequence that repeated and repeated and repeated over time. We know that that induces plasticity in the left primary motor cortex as people learn that task. And so the question was, in that context of plasticity induction, can we see decreases in GABA? I put this up because it's because I know it quite well, but also because it illustrates some of the issues that are really important when we're trying to study plasticity in any context. So we've got our condition here, the first group, where they were indeed learning this motor sequence learning task, and unsurprisingly, people are capable of learning which button comes next in a sequence task if you pay them enough and put them in a scanner for long enough. But we were interested in seeing decreases in GABA, and the first 3 talks this afternoon will have hopefully given you a few ideas as to why you might see a change in GABA that has absolutely nothing to do with whether or not people are inducing plasticity. Okay, so we were worried that you might see a change in GABA if you get frequency drifts, if you increase movement by, doing some movement, and you decrease the quality of your scan. So it's really important to have a condition that is matched as closely as possible to the one that you're interested in, to the learning condition, in every respect apart from the fact it doesn't have the learning component, okay? So in this, exactly the same condition, but instead of there being a sequence to learn, there were some random button presses. There was also a time in the scanner condition where people just watched a David Attenborough nature film and didn't do anything else to make sure we weren't being biased by any overall drifts in the data. And what we saw was, luckily, which is why we're here, that we, over 6 blocks of 5 minutes of spectroscopy, our GABA measurement decreases in the left primary motor cortex as subjects learn the task, but not when they're just moving their fingers or when they're just lying at rest, trying not to fall asleep. Okay. So we have a specific decrease in GABA in the brain region we think is doing the learning during learning of that task. Can we go any further with that? Well, we can start to look at relationships between GABA levels in that first block and see if they predict how much people learn on a subject-by-subject basis, and indeed they do. And we can start looking at neurochemical specificity, and we can do exactly the same analysis and say, is this just true of inhibition excitation in the brain? Is this just something to do with how much neurotransmitter there is in general? Or is this something very specific about the amount of inhibition that's in the brain? And so we tend to use glutamate as our neurochemical control, because it seems like quite a sensible one to us. And we see a relationship here whereby GABA concentration early on a subject-by-subject basis predicts how much you learn, but that is not true of glutamate. Okay. So those kind of studies have been done a few times and have found very similar things. As I say, motor learning is a really easy way of looking at learning because you can measure the amount of learning really easily. You can get reaction times and they behave really nicely. But people are interested in brain areas outside the primary motor cortex. I stand here as somebody who's not quite sure why, but apparently people are. And so one of the big questions we might have is, okay, there's a lot of evidence that GABA might be important for learning of motor skills, but do we think it might be important for learning of things that aren't motor skills? And this might be particularly relevant in primary sensory areas where it's at least plausible that inhibition might interact with learning in different ways. So this is work done by some colleagues of ours in Cambridge, by Polly, who is there, so please feel free to ask her if there are any technical questions, that they very kindly did in Oxford, and we had a lot of fun doing it. And the question they really had, they're a vision group, and they were very interested in the relationship between GABA and learning of various perceptual tasks. And being very clever, they had 2 tasks, and they had very clear hypotheses about what should happen to the GABA. So these are the original prototype stimuli, and the task, as far as the subjects were concerned, was to see these and say whether the dots were basically aligned radially or concentrically. But they mixed them up a bit, and they made them quite difficult to do. So there was one task where they took those stimuli, but they mixed it up with a lot of noise. And the hypothesis there would be, as people improve in being able to distinguish between these 2 types of stimuli, they might do that by decreasing their GABA levels, because you want to get all the information you possibly can to allow you to pick out the relevant stuff. They also had a feature differences task where they were very interested in looking at very specific differences between these. And they had the hypothesis there that in order to distinguish important features that might allow you to tell which type of stimuli it was, you might want to put your GABA up, okay? Reduce the noise and look very specifically at the signal. these are the behavioural results. They do a lot of trials. This was not a fun experiment to be a subject in. We're very, very grateful to them all. And you can see over time people get better at being able to correctly identify these stimuli, so they can learn it. This is a particularly nice experiment because not only did we have a control neurotransmitter, but actually they had a control voxel. So we did simultaneous acquisitions where they were interested in lateral occipital cortex, which was where we thought this learning would happen, and in the posterior parietal cortex, where as a site that is involved in this kind of signal processing, but wasn't hypothesized to be the site at which learning should occur. And these are the results. What they saw was exactly what they predicted, such that in the signal-to-noise task, where you might imagine you want to decrease your GABA to maximize the information available to you to make that discrimination, we saw a significant decrease in GABA, whereas a condition where you might want to increase your GABA to reduce that noise and really look for the salient features, there was a significant increase. That split was specific to the region that they hypothesized it might be. But we did see a sort of increase in GABA in the other voxel. We often see that. I'm very interested to discuss and get a view on what people think that might be. I wonder whether it's alertness and attention in the cortex. In the scanner, it's a pretty dull environment, even doing this kind of stuff. And these changes were indeed specific to GABA. There was absolutely no change in glutamate in the occipitotemporal cortex. They went on to look and see if this, again, like in the context of the motor system, whether or not we could relate the change in GABA to the behavioural improvement, with the idea that if this really is doing something and allowing us to learn these tasks, then you might imagine that people who can decrease their GABA a lot are going to do really well at learning it, which indeed they do, and people who really aren't doing very much down here really aren't learning it very well. And the interesting thing was we saw that increase in GABA in the feature differences task. It's difficult to know whether that's just a time effect or whether that's a task effect, but speaking very strongly to the idea it's a task effect, there is a highly significant relationship in the other direction, such that in this task, where you want people to put their GABA up to get better, people who do get a lot better, people who don't or even decrease their GABA actually really don't learn. So this is not a phenomenon that's restricted to the primary motor areas. It seems to happen in primary sensory areas. But we shall see whether this works. Learning is not just restricted to the period of time in which you're doing the task. Right? So anybody who's learnt anything, so if you've learnt to play a piano piece, you will know that you learn it and you kind of get okay at it. And then you go away and you come back and you're better at it. Okay? So we have a learning phase and we have a memory stabilization phase or consolidation phase, where things that you have learnt then get stabilized and those synapses get stabilized. And so the question then is, what happens, do you think, to GABA during memory stabilization? Does it carry on decreasing? Does it increase? Or does it stay much the same? And I have one, well, either everybody's right. I haven't got an N on this. I'm going to assume that you're all very intelligent and paying attention, because that is entirely right. Thank you very much. Can we go back to the slides, please? So yes, absolutely. You might expect that if you get a decrease in inhibition that allows these synapses to become quite labile, once you've got the right synaptic activities, they then get consolidated, stabilized by an increase in inhibition. So this was work done over in the States by Watanabe's group in a really neat design. And what they did was another visual task, where task one, they asked people to try and work out what angle there was on a visual grating. And it was very difficult to see the visual grating, so people concentrated on it and learnt it. But they got better at doing that. They then waited a little bit and did another task that was extremely similar. Okay? And the idea being that if you do two tasks, if you learn one thing and then you learn another thing in close succession and they rely on the same brain mechanisms, then learning of task two will basically remove the learning of task 1. And they looked at that by looking at the how much memory there was on day 2 compared with how good people were at the task before they did it. They had another condition where they spent much longer learning task 1. They called this overlearning. So they learnt it and learnt it and learnt it to the point that the behaviour had stopped improving, and then they carried on. So they stabilised that memory and did exactly the same contrast. In the condition, as you would expect, with no overlearning, if you look at the behaviour improvement here compared with beforehand, you, on the first train session, you actually get a decrement, no real change, because you've wiped out this learning with learning of the second task. But in the overtrained condition, behaviourally, they demonstrated that they had stabilisation of that memory, such that even if you then learn a second task, you don't learn it very well, and actually that first memory is maintained. Okay. So we've got 2 conditions whereby that memory, the memory of the first task has been lost completely, and the second one where that memory trace was stabilised before the introduction of the second task. So we've got stabilisation of synapses. So they did exactly the same thing and just looked at Gaba levels before and after, either just learning the task or really overlearning and stabilising that intervention. This is the normal condition. One of the delights of the Gaba literature is that everybody plots it in different ways. So this is an excitation-inhibition balance. The excitation doesn't really change. So an increase in this basically means a decrease in Gaba. So this is a decrease in Gaba. This is an increase in Gaba. And if you learn a task, as hopefully you are not surprised by now, you see a decrease in Gaba. However, if you've overlearnt that task and you've stabilised it, and they know they've stabilised it behaviourally, they see an increase in Gaba representing that stabilisation. Okay. So it matters what stage of learning you're looking at. Are you looking at the short-term acquisition and the active acquisition of behaviour, or are you looking at what happens after that as there's no behavioural improvement, but you go to stabilise and consolidate that information? So that was looking at short-term learning, but looking at what happens after you've learnt it and looking at consolidation of that learning. The other situation that we see a lot and we might be interested in is what happens to neurochemicals during longer-term learning and plasticity. Most of the skills that we're actually interested are not things that we learn over half an hour, are things that we learn over days, weeks, months, and years. And so work done in Oxford by colleagues of mine looked at learning of a complex visuomotor skill over some weeks by teaching undergraduates to juggle. Okay. This was a lot of fun. They had a lot of entertainment doing it, but actually it is, if anyone has learnt to juggle, it is a complex skill, but one that pretty much anyone can acquire if you do enough of it. This was the experimental design. They did a baseline session, we did lots of imaging, including spectroscopy. They then went away and trained for 15 minutes a day for 6 weeks and got better. They then came back, had another spectroscopy scan, had 4 weeks of not juggling, and then had another scan. This is the behaviour, so these were ratings, and you can see that people, we're interested particularly in the low intensity group, I won't go into the high intensity group here, got better over these training days, and they got, most of them learnt to juggle pretty proficiently actually. These are the Gaba levels, and what you can see is at baseline, and then you get a decrease in Gaba during the learning. What happens though after you stop learning is that it goes back to baseline. Okay, so you get this decrease in learning, decrease in Gaba during active learning, but once it has that learning has actually finished, and you have a short period of consolidation, probably that's a few hours, and then if you don't use that skill again, actually, as you might imagine, your inhibition returns pretty much to baseline. This is still a very, very open question, though, because there is also some work done by Jakob Blecher, who was interested in relearning of motor skills after stroke. So if any of you have seen a patient who's had a stroke, people often lose a lot of function and will recover that function over days, weeks, and months and years. And so we can think of it as a sort of in vivo clinical way of looking at long-term plasticity. And so he did exactly this, and he looked at GABA levels in the affected primary motor cortex in patients with subcortical strokes, and just asked the question, if this is a marker of long-term plasticity, do they have lower GABA levels than people who haven't had a stroke? Which is exactly what he saw. So this is not due to the fact that there's a lesion in this voxel. There isn't, and they have corrected appropriately for atrophy and other measures. This is a genuine decrease in GABA levels in the patients, hypothetically, representing a long-term induction of plasticity to support motor skills. Why do you see a difference between the juggling study and here? I don't know. It would be a very interesting study to look at. We would suggest, and I would suggest anyway, that the patients here are still learning. They're still improving. It may be quite slow by these points. These are months or years after their stroke, but they're still learning, they're still actively engaged, and there are still active plasticity induction and plasticity maintenance mechanisms occurring in their primary motor cortex, whereas in the juggling study, that has been finished. They're no longer allowed to learn it, and therefore things have returned. But that is a hypothesis that needs testing. Okay. So for the last 5 minutes or so, I'm going to talk to you a little bit about one of my interests, and something that I hope might spark a little bit of interest in a few people here, which is the use of non-invasive brain stimulation approaches to determine causality. Okay, so non-invasive brain stimulation is a useful way, we could do this by a show of hands, by which we can get around the fact that MR is inherently a correlational modality, and that's true for anything you'll see in the next few days. And really, if we want to know whether something is causal, we need to interfere with it and see what effect that has. Ooh. That's exciting. Yes, excellent. Okay. And you can do that in a number of ways. But one of the appealing ones in humans, anyway, is non-invasive brain stimulation. People do this in a number of ways. The work I'm going to talk about in the next two studies is using transcranial direct current stimulation, whereby we apply a 1 milliamp current to the brain region. Oh, excellent. I know about this. This is brilliant. I like this. When it works, this is good. We apply a 1 milliamp current to the brain region that we're interested in to see, to manipulate ongoing activity there and see what effects that might have. Great. So if I can go back to the slide. So we use transcranial direct current stimulation. There are lots of other things to do. This is the montage that we use. We have an electrode over the left primary motor cortex and a reference electrode. And we were very interested in whether or not inducing plasticity exogenously by using non-invasive brain stimulation might have similar effects on GABA to inducing it endogenously by things like learning. And indeed it does, such that if you use anodal TDCS, which we know induces LTP-like plasticity in the underlying cortex and is an excitatory stimulation, we see a significant reduction in GABA after about 10 minutes of stimulation of about, in the order of about 10%, which is about what we see when we do a learning study. So that's great, and we've run with that, and we've done lots of brain stimulation studies that I won't bore you with. But one of the things we thought about and had been bothering us for a while was we see this GABA decrease with learning, but can we get any way to saying that we think it might be causal or whether it might be an epiphenomenon. And one of the ways then we thought about doing it is a study in some young healthy people. And what we did was do anodal TDCS, the left primary motor cortex, on one day and measure on a subject-by-subject basis how responsive their GABAergic systems were, so how much their GABA decreased in response to stimulation. We did a control voxel, which showed nothing, as you would hope. And then on the third day, we did a motor learning task, zero reaction time task, as before, during functional imaging. And this design allowed us to ask the question, can we relate on a subject-by-subject basis how good people are at learning to how much their GABA decreases when we induce plasticity? Okay. And if GABA decreases are this sort of gating mechanism, then there should be a specific relationship between those two things. So we calculated our learning measure just by the decrease in reaction times, as we usually do. And what we saw, as you might imagine in this talk, is a significant relationship. So people who were had very responsive GABAergic systems for whatever reason were also those who learnt quite well. People who didn't really modulate their GABA didn't really learn very much. So it's not causal, but it's a little way towards suggesting that this might be an important mechanism. And you can look on the fMRI signal, and actually that relationship between the learning-related fMRI signal and the MR spectroscopy decreasing GABA is very, very specific to the left primary motor cortex, where we think that learning is happening. So it's a way that we might start to actually relate these things. I'm going to finish with what is a slightly complicated study by colleagues of mine in Oxford, but is a very, very clever way of looking at the role of GABA in learning. What they did was get people to scan it and learn these associations. So they asked people to learn that if you saw stimulus A, you would expect the next stimulus to be stimulus B. Okay, and if you learnt C, you'd expect the next one to be D. So they learnt these pairs, arbitrary pairs of stimuli. And they relied on a property of the brain that is that it gets a stimulus repetition. So it gets bored if it sees the same thing twice. And if you really learn these pairs, then the activity you expect to see on the second stimulus is much less if the preceding stimulus is the one that pairs with it. So if you have A and then B, the brain knows that it's expecting B and doesn't get very excited about it, and the bold response is quite small. If you have A and then C, the brain isn't expecting it, and you get a much bigger response. Okay? So it's a slightly weird way of looking at learning, but it allows you to quantify how well learnt these associations are. So they did a very complicated design where they did exactly that. They trained these associations, and then they looked at those responses, and what they saw was that you get this difference in response to the expected stimulus early on when you've just learnt it. But actually, once, like the overlearning condition, once you've learnt it for a while, actually it goes away and there's no difference. If you imagine that we get an increase in excitability in our brains every time you learn something, we're going to be all over the place. So this then disappeared. What they then did on the second day was test this again and showed, and then did anodal TDCS to decrease the inhibition, which indeed it did. And so what they then saw was that on day one, you have this learning trace, and actually that then disappears. Okay. It's still not there on day two. You've learnt it. There's nothing going on, much like the juggling study. They've learnt it. They're not learning anything anymore. The fMRI signature of learning has disappeared. But what they then did was use anodal TDCS to decrease the GABA, and suddenly this learning trace reappears, and the degree to which it reappears is related to the GABA. So what they took from this is that not only are there glutamatergic traces, there are new synapses being formed in glutamatergic synapses that support this learning, but that actually after that, there are also anti-memories, so similar circuit changes happening in the Gabaergic neurons that then renormalize the excitatory and inhibitory balance. But then if you get rid of those by decreasing the GABA, you get that re-emergence and revisibility of the glutamatergic memories. So I hope that I've persuaded you that either GABA is important or GABA changes are important for plasticity and learning in humans in a range of situations. And MRS, if used carefully, and we've heard a lot about the things you might want to think about when designing experiments, can tell us about human plasticity across multiple regions, domains, and timescales. And if you combine it with other imaging modalities, behavior, or invasive methods, we can really start to get a little bit closer towards examining causality. Thank you very much.
So there's one question that had 2 votes. Do you think those with more experience, 3 votes now, do you think those with more experience in learning, like university students, would show more of a change in GABA in those tasks? That's a good question.
It's a really interesting question. So we do get predictions between GABA levels at baseline, how much you'd learn. So we think that people, so I would hypothesize wildly that people who have very responsive GABAergic systems in the motor cortex are probably going to be good at learning all kinds of motor tasks. What seems to not be clear is that that translates across modalities. So the fact that you're really good at learning a motor task doesn't mean you're really good at learning anything else. So we do often do our experiments in our undergraduates who are quite bright. We don't often tell them that, but probably quite good at learning stuff. But these do seem to be fairly consistent in slightly more representative populations.
Thank you very much. We'll leave the other questions for after the talk. Thank you.
Great. And it gives me great pleasure to introduce Nick, who, beyond spending the last 6 months cajoling us all to actually put our presentations together and arrive in Rome at the right time, actually also finds some time to do some research. He's an assistant professor at Johns Hopkins and does a lot of seminal work on spectroscopy and clinical disorders. So thank you very, very much.
Thank you very much. So in the interest of time, I only have 25 minutes. I'm going to focus on GABA and talk about GABA in psychiatric and neurological disorders. Again, I'll use some of these features, so please log in now. And actually, the next question is going to be a poll that I'm going to see if it works. So the question is, who is currently working with clinical populations, who's interested in working with clinical populations, and who is focused on basic science? Do you know if it will come up? Right, maybe show of hands. Who's interested in working with clinical populations, or currently working with clinical populations? Who is not currently but is interested? Okay. Pretty good. All right, we can go back to the PowerPoint now. All right, so we got a nice range of people. So the goals of my talk is for you to understand in which disorders GABA is thought to be altered and how these alterations might occur, to understand how spectroscopy can play an important role in understanding neurological and psychiatric disorders, and to understand how MRS can and cannot be used for diagnosis and treatment development. So I'll first talk a little bit about GABA, refer back to Professor Rae's talk in terms of where things might be going wrong. I'll then talk about considerations for designing your clinical studies rather than just jumping out and talking about 25 different disorders. But I'll use some disorders where GABA is altered to point out these considerations, and I'll talk about age and development as well. I'll then spend some time talking about pharmacology and some of the limitations of working with these populations. So we know that GABAergic transmission plays a key role in healthy cortical functions, healthy behavior, and cognition. And so when GABA isn't functioning properly, that can really lead to large repercussions and therefore to psychiatric and neurological disorders. It is widely distributed in the brain, has important roles in inhibitory excitatory balance, synchronization of cortical activity, as well as plasticity. And there's a large variety of GABAergic drugs on the market that we know work in a variety of different disorders. And I put GABA here in quotation marks, because as Professor Rae mentioned, these are wide-ranging drugs. They don't point to a specific GABAergic function, and some don't even function on GABA at all. They just kind of secondarily affect the GABA system, and so it's important to keep that in mind. But these drugs do seem to work, and sometimes people are less interested in the actual mechanisms of the drugs, in the fact that they do something to the patient that makes their lives better, which is ultimately what we were trying to do. In addition, repetitive TMS is a technique that's being increasingly used to target specific aspects of disorders, particularly depression currently, and I think that has some effects on the GABAergic system as well. The thing I would like to point out right now is that MRS of GABA is currently not being used for diagnoses. All the studies I'm talking about are research studies. It's currently not being used in the clinic. So a couple of years ago, Richard and I reviewed the GABA MRS literature, including focusing on kind of clinical applications. And we found then that the majority of clinical studies looking at GABA at that point were focused on depression and related disorders, as well as schizophrenia and epilepsy. And then quite recently, Schur and colleagues did a fantastic review where they also did a meta-analysis of the results found in papers, but they really focus on a number of different disorders, including major depressive disorder, schizophrenia, and autism, as well as some other ones. Another area of interest that has been studied widely is pain disorders as well. And so there's a wide body of research focusing on these different disorders, and if you want to know more about specific disorders or what disorders have been studied, I would point you to these reviews. But I first really want to talk about where things might be going wrong in the GABA system. And as Professor Rae said, the GABA system is a very complex system. What we're measuring is all the GABA within this huge predefined region of cortex, and so we can't differentiate where our GABA deficit is coming from. This could be, for instance, due to macroscopic drivers, such as differences in cell density or differences in synapse numbers, but it could, for instance, be in terms of the production, breakdown, and receptor function of GABA. So it could be, for instance, in glutamic acid decarboxylase, or GAD, which has two predominant isoforms, GAD65 and GAD67, that affect the production of GABA, and so an issue there might lead to differences in GABA. However, it could also be in the transport of GABA, GAT might be affected, or GABA aminotransaminase might be affected. That leads to differences in the breakdown, the transport and the breakdown of GABA. And finally, GABA might be, or the GABAergic system might be affected in terms of GABA receptor function, as Professor Rae nicely outlined. And there, the relationship to that and the overall GABA as a neurotransmitter that we measure is perhaps less clear. This probably is a relationship, but it's quite complex and not as direct. And so the issues could be in terms of GABA production, could be in terms of GABA transport, which is more kind of presynaptic, astrocytic, or GABA receptor function, which is more in the synapse and postsynaptic. And it is possible that there's disorders where you have a collection of patients which have different fundamental underpinnings. There could be people with differences in GABA receptors or differences in GABA production that show very similar phenotypes. And this could happen, for instance, in disorders like autism or schizophrenia, where there's a wide range and a lot of heterogeneity between disorders. Having said that, with MRS, we do measure differences in the amount of GABA that we have, which does point to differences in neurotransmitter, or at least GABAergic availability, which might have large repercussions on cortical functioning and behavior. And so it does allow us to tell us something about the GABAergic system and potentially look at diagnosis or treatment. So here's a poll. What do you expect to see in a patient who has antibodies against glutamic acid decarboxylase? See if this works. And we've heard this mentioned a couple of times now. All right. Does GABA go up? Does GABA go down? Or do you see no difference? I've got one vote so far. 2 votes. All right, 5 votes. Okay, we can go back to the PowerPoint now. Some adjustments, but most people talk about it that way. So there's actually a disorder called stiff person syndrome. And stiff person syndrome is not very common, but it's characterized by muscle spasms, muscle rigidity, as well as epileptic seizures. And it can mostly be diagnosed by titrating for glutamic acid antibodies. And about 65 to 80% of patients have antibodies against glutamic acid 65, which means that that enzyme isn't working properly and is therefore not producing GABA properly. And so in this study, they used 2DJ spectroscopy in a small group of patients and controls, and they measured GABA in both cortical regions as well as subcortical regions. And what they found was that the GABA levels were substantially reduced in the patients to about 40–50% in these patients, particularly in the cortical regions in the parietal lobe, but not in the subcortical regions or the frontal lobe. And so here what we're seeing is that we know that in these patients Gas65 is affected, therefore affecting production of GABA, and we can measure that difference in production of GABA using magnetic resonance spectroscopy. So I want to use a number of disorders to talk about considerations for designing studies and what you might be able to learn from working with patient populations. So I first want to talk about the specificity of measurements, talk about neurodevelopmental disorders. I then want to talk about the heterogeneity of disorders as well as establishing the right outcome measures in disorders, and do that with respect to depression. I want to talk about function and behavior with respect to borderline personality disorder, and establishing a clinical trajectory with respect to schizophrenia. And then I'll talk about development and pharmacology a little bit. So my own work focuses on neurodevelopmental disorders, so I'll talk about that first. So I work with children with autism spectrum disorder, children with Tourette syndrome, as well as children with attention deficit hyperactivity disorder. And obviously these are very distinct disorders with their own etiologies, but there are some behavioral commonalities between the disorders. Now, what is common between these disorders is that there's quite a wide body of evidence suggesting that GABA is altered in these disorders. And so we scan children and we put them in the scanner, we measure the GABA levels. And so in autism, we found significantly reduced GABA levels in the sensory motor cortex of these children. We were absolutely not the first to find this, we were not the last to find this. Seems to be quite a consistent result, where you see reduced GABA levels in the motor cortex or sensory motor cortex of children with autism. What is also quite consistent and very interesting is that visual GABA levels do not appear to be affected in autism, and I think that is something that I'm happy to discuss afterwards why this might be. But that is also a consistent finding, so there does seem to be some regional specificity to these findings. However, we also see reduced GABA levels in children with Tourette syndrome, and in a previous study we also see reduced GABA levels in children with ADHD. And so reduced GABA levels per se in children isn't an index or provides diagnostic information about the disorder. We can't just put an 8-year-old child in the scanner, see reduced GABA levels and say, well, this child has autism. It's also not really relevant because autism is diagnosed at 2 or something like that. And so it's not really relevant. What becomes more important is that we learn something about the behavior outcomes. And so we don't see any specificity in these disorders. We know these disorders are heterogeneous, so this kind of average GABA levels, how meaningful is it? What we really need to look at is, are there relationships with any clinical features that are informative? Now, in our results, in all these studies, we do actually find very specific behavioral deficits relevant and specific to the disorders that GABA correlates with, but I actually want to talk about that with respect to depression. So the GABA and depression has been a long interest, and in this study, GABA was measured in the anterior cingulate cortex of youth with depression. But in this case, they separated out youth with depression with anhedonia and without anhedonia. And anhedonia refers to an inability to feel pleasure, which is one of the comorbidities of depression. And so they did this in quite a large subject number, so 44 people with depression and 36 controls. And what they found was that ACC GABA levels were significantly lower in depressed youth with anhedonia, but they weren't significantly reduced in those without anhedonia. And so here you have a disorder where you have a subgroup that is affected in terms of GABA levels and a subgroup that isn't. And in disorders that are heterogeneous, you need to be very careful that you don't pull all these data together necessarily, because you might not see a difference, because it's really different by subtypes that are slightly different chemical subtypes or different chemical mechanisms underlying these. In addition, they also found an important correlation. So here on the x-axis you see GABA concentration, on the y-axis you see anhedonia score. And it was those participants with higher GABA levels who had less anhedonia, so were less affected in terms of the disorder. Whereas those participants with lower GABA were more affected in terms of the disorder. And so here you really start stratifying a disorder in terms of GABA levels, where GABA levels point to a very specific processing deficit within this disorder. Again, choosing your symptoms and outcome measures here is very important, because if you look at maybe a generalized depression metric, you might not see these differences. But with anhedonia specifically, knowing that kind of that behavior is also related to ACC processing, you do see these differences. And so choosing what symptom you're interested in might provide you more power and more interest for looking at these disorders. I also want to talk about the study with borderline personality disorder, which is predominantly identified by impulsive behavior as well as sensation seeking. And in this study, they measured GABA levels in all female cohorts of patients with borderline personality disorder, or BPD, as well as a matched healthy control participant group. And they used suppress measurements. So they looked at, they actually suppressed the macromolecules to get a more pure GABA measurement. And they found, they also did an fMRI task where they look at interference inhibition, which is a measure of inhibition and impulsivity, but more approached cognitively. So they found lower ACC caudate functional connectivity, both in the ACC and in the caudate, in these BPD patients. Now, in addition, they found that in these patients only, anterior cingulate cortex GABA levels correlated significantly with the ACC and caudate bold activation, as well as the functional connectivity between them. And so what they have here, they started linking the neurochemical level with the functional level. It becomes more interesting when they start looking at clinical measures. So they found that both GABA and ACC caudate connectivity, as well as the caudate activity as well, significantly correlated to this a clinical measure of sensation seeking. And so then what you can then do is do a mediation analysis, because they found that both GABA and bold activation related to this measure of sensation seeking, but they're also related to one another. And so they found that the relationship between GABA and this behavioral measure of impulsivity was significantly mediated by the bold activation. And so what you can start doing here is you start getting much more of a mechanistic idea of the system that you're working with, because you don't only have the kind of cortical functional connectivity measure, you actually have to what extent does the neurochemistry that underlies this relate to both the connectivity as well as the behavioral outcomes. And so you get a much more of a full picture of a mechanism in a disease, and maybe more points in which you can start interfering, providing treatment, or at least tracking this disease itself. Schizophrenia is one of the most widely studied disorders, and the GABA glutamate hypothesis of schizophrenia is very prevalent. There is this idea that there is glutamate excited toxicity in the brains of people with schizophrenia that leads to neuronal damage. Also, too much glutamate leads to inefficient GABAergic function in these patients, and there's also been an implication of glutamic acid decarboxylase, the 67 isoform in schizophrenia. So in this study run by Roland and colleagues is very important because it was one of the largest studies done in schizophrenia, and I'll get back to sample sizes in the limitation section, where they measured GABA in the anterior cingulate cortex in a group with an older group with schizophrenia and a younger group with schizophrenia. And they separated the analysis. And they found that GABA levels were significantly reduced in the older group with schizophrenia, but not in the younger group with schizophrenia. In addition, they did a linear regression where they found that there's an increased decline in GABA in patients with schizophrenia compared to typically developing controls, where GABA levels decrease slower with age. And so what you start looking at here is a trajectory of a disorder, where early in the disorder you might not see differences because the disease hasn't progressed yet, but later in the disorder you might. And so understanding the clinical trajectory of a disorder is incredibly important, because this really leads to understanding outcome measures, the progression of a disorder, when you can intervene, etc. And so this study is important for a few number of reasons. One is they use a very large cohort of 145 patients, which is very important. Secondly, they use this macromolecular megapress sequence, but they replicated their results of lower anterior cingulate cortex GABA levels in older patients in a study from 2012 where they measured GABA+. And so they used two different techniques here, but they found a consistent result in terms of reduced GABA levels, and I think that is also very important. But the most important difference here is that they found a difference in the age trajectory between patients. Now, what is more important is, can we actually identify a disorder before onset of kind of active symptoms? And so in this study, they measured cortical GABA levels in subjects at ultra high risk for psychosis that they measured using a community assessment tool for at risk for psychosis. And in 20 patients at ultra high risk, as well as 20 healthy controls. And they measured GABA in a frontal voxel because that's been associated with psychosis before. Now, they didn't find any significant differences in GABA levels. This is consistent with the previous study by Roland and colleagues, but in the younger cohort with schizophrenia, they also didn't find any significant differences. However, they did find a difference in terms of the relationship to this measure of at risk for psychosis, which is separated in positive symptoms and negative symptoms, where more negative symptoms is more likely to end up in psychosis. And they found that those participants with higher GABA levels had fewer negative symptoms, so fewer prodromal symptoms for psychosis, where those participants with lower GABA levels had higher, were more at risk for psychosis. And so here you can start looking at the early trajectory of a disorder, which is incredibly important because early identification of a disorder leads to early intervention of a disorder, and pretty much in all disorders leads to better outcome measures. And so being able to identify a disorder very early in its development, or even before onset, when onset might occur, is very important for targeting treatment and targeting behavior. GABA and glutamate and other MRS measures have been widely described in schizophrenia. So obviously, I don't know, I could spend 2-3 hours just talking on schizophrenia alone. But I would say that there's 2 excellent reviews out there, one by Gerard Dwyer and colleagues, and one by Jeroen de Jonge and colleagues. And if you're interested, I would look these up further. So aging and development is important, and Dr. Harris talked about aging and development somewhat. So the results on aging and development are very wide-ranging, they're not consistent, and studies use different techniques, different sample sizes, etc. But they also use different cohorts. And so together with Eric Porges and Greg Jensen, we recently did a meta-analysis of the data on the age trajectory in terms of GABA levels, where we looked at all the study that had a wide enough age range where the individual data points were plotted. We extracted these data, and then we normalized them first within group and then across all the groups, and then looked at the age trajectory. And we used a number of different models to see what best fits this age trajectory, but we found that the log normal fit fits this best. And so what we see when we combine all these studies and therefore massively increase our power in terms of the data that we have access to is that GABA levels increase very significantly, very quickly early in development, but then taper off around adolescence and early adulthood and then have a slow decrease. Now, it should be taken into account, as Dr. Harris said, that there is a tissue component to this effect, and some of these data were tissue corrected and some were not. So this is definitely a limitation. But this does seem to be the trajectory that is most consistent and is consistent with other age trajectories in terms of cognitive function. Now, what I should mention is there's a huge gap before 6 years old. Scanning children before 6 years old is difficult, but we're trying to do this. And I think that is important to see what happens very early in development, because we know that GABA plays a particularly important role in early development. Now, in the last bit of my talk, last couple of minutes, I want to talk about pharmacology, because obviously pharmacology is very important in treating disorders. And so the first study I want to talk about is gabapentin. So gabapentin mimics the action of GABA, but it doesn't actually bind to GABA receptors, it binds to calcium channels and therefore mimics that action, and is most often used in the treatment of epilepsy, anxiety disorders, as well as migraine. Now, in this study, which is one of the earliest studies using editing to look at GABA in patient groups, they gave gabapentin at 2 different doses to groups of patients with epilepsy, and they compared their GABA levels to a group with matched epilepsy drugs that weren't particularly GABA-focused, such as valproate. And they found that the GABA levels in patients who get gabapentin were substantially higher than in those who are not on GABAergic drugs. In addition, the number of seizures decreased substantially in the patients, particularly on the high dose. And so here, you can start tracking the response to a treatment on GABA by measuring GABA using magnetic resonance spectroscopy to see whether GABA levels themselves change within the brain. However, this doesn't always work. So in another study where they looked at the GABA response to acute benzodiazepine administration in panic disorder, they gave patients with panic disorder as well as comparison subjects benzodiazepines. So in this case, it was clonazepam. And benzodiazepines increase the efficacy of GABA receptors, but there's also this idea that it actually affects the glutamic acid decarboxylase, so affects the production of GABA. And so what they did here is they gave these patients benzodiazepines and measured GABA levels acutely after giving them these drugs. And they found in the comparison subjects that GABA levels on average, can't see it. So in the comparison subjects, GABA levels on average went down. And GABA levels also at baseline were actually higher in the controls than in the patients with panic disorder. However, in the patients with panic disorder, benzodiazepines didn't actually affect the GABA levels. It did nothing. And I think this is very important because it shows that when you have an abnormal GABAergic system, drugs that affect GABA might respond abnormally compared to patients. You can't always expect that your system is working the same in a population where that system is altered. And so in this case, the patients didn't respond. And non-responders in terms of drug treatment to different disorders is a huge issue, because you always want to know, is this person going to respond, yes or no? And so perhaps based on GABA levels, we can start predicting whether our people are going to respond to GABAergic drugs or people aren't going to respond, which is substantially going to improve the treatment of these disorders. So we heard about vigabatrin a little bit, so this should be a question. So vigabatrin is an inhibitor of gamma aminotransferase. So if you give that to patients or to participants, what would you expect to happen? I'll try again. Right, we'll do hands. If you block GABA transaminase, who thinks that GABA goes down? Who thinks that nothing happens to GABA? Who thinks that GABA goes up? Right? Some people, that's the main response. All right, got 3 responses there. Okay, can we go back to the slides, please? All right. So here was a study where they gave vigabatrin to people with epilepsy. So vigabatrin blocks gamma aminotransaminase, which makes succinic acid from GABA and breaks it down. And so when you block the breakdown of GABA, you expect that GABA starts accumulating in the environment. And so that is exactly what they found. So they gave these patients different increasing doses of vigabatrin and found that up to 60 milligrams per day, the GABA levels went up in the brains in their brains compared to their baseline measure before vigabatrin treatment. In addition, what was very important clinically is that their numbers, the time since their last seizures went down substantially, and so there was much more time between seizures. And I think that is also an important thing to keep in mind, is that you can study the GABA system or give drugs, but ultimately what really matters is the outcome measures in these patients. Do they feel better, and do they respond to the drugs in a clinically relevant way? So in this case, a drug purposely led to increased GABA levels and accumulation of GABA levels by blocking the breakdown of GABA. But there's another very rare disease called succinic semialdehyde dehydrogenase deficiency. In this case, GABA is broken down to succinic semialdehyde, but doesn't get broken down to succinic acid. It's blocked in that process because it doesn't get broken down any further. And so what happens is that you can start accumulating GABA in the environment. In addition, what happens is that through semialdehyde reductase, GABA starts being broken down into GHB, which is more commonly known as a date rape drug or as liquid E, which is really not something you want to have accumulating in your environment. And so these patients suffer from huge issues. So there's developmental delays, cognitive delays, seizures, etc. And so in this case, they did unedited spectroscopy, I would have to say, in the gray matter voxels in patients with this, in one single patient with this disorder. And they found that both GABA and GHB, which you can also measure using spectroscopy, accumulated substantially up to 300% in the voxels. And so here in this case, accumulation of GABA is a bad thing, and accumulation of GABA and too much GABA, as well as GHB, leads to neuronal damage, as well as huge cognitive effects. All right, so based on all the talks so far, what do you think is the most important factor when studying, when looking at patient populations and seeing reduced GABA levels? This works. All right, who thinks motion is the biggest issue? Who thinks motion is the biggest issue? All right, who thinks medication is the biggest issue? Who thinks, you can go back to the slides, who thinks that gray matter composition is the biggest issue? One vote there. And who thinks that age matching is the biggest issue? Right, so I think that so all of the questions would have been right, but it depends on your population. So for me, working with pediatric populations, and particularly pediatric populations with, who are hyperactive, often motion is for me definitely the biggest issue, because these children just move more. And we know that increased movement leads to reduced GABA levels. And so when I see reduced GABA levels, I need to make sure that there are no differences in data quality between these groups, or that there's not any outliers driven by poor data quality due to movement. Now, medication in other disorders is a huge issue, and the same for the disorders I study, because medication, as you have seen, can substantially affect the GABA levels in the brain. And so there are populations where, for scanning them, you might take them off medication. Now, sometimes these medications act acutely and they wash out very quickly, but there are medications where, even if they don't take it for 2 days, you still see a chronic effect. There's also populations, for instance, pain disorders, where you just can't get people off their medications because they suffer. And so medication needs to be taken into account when you're studying these different disorders. The same as Dr. Harris talked about, tissue correction, and tissue is a huge issue, but sometimes you are studying populations with a lot of atrophy, and so then you need to take into account that there is less gray and white matter in these voxels, and so you need to account for that. So age matching is actually something that you should be doing, because as we've seen, age can have a substantial effect on GABA levels, and if age is not the thing that you're interested in, you should age match and account for age in your analyses. So there's a couple of limitations I do want to talk about, and these limitations, and it's important to take in mind that some of these limitations that I just spoke about affect the actual GABA concentration in a neurochemical way, and some affect the GABA measurement that we have because they affect the quality of the measurement. So one thing that affects the quality of our measurement is scan quality and motion, and I think that is something that you need to look at. This is something you need to report, and this is something that you need to account for. And something that can substantially help with this is, for instance, training of subjects in the scanner, doing mock scans, taking them through with children. A story helps a lot. Sample size is a huge issue in clinical populations. I pointed this out before, but it is tricky because scanning clinical populations is difficult. Access to participants is difficult. So there's a lot of studies that do suffer from low subject numbers. So this is from the Schur paper, and you can see that a substantial number of studies have subject numbers within each group with less than 20 subjects. And so if your effect size is big enough, sure, you probably can see your differences there, but you might not be able to see differences when you have a heterogeneous but small sample size. You see this in an IRAM paper, which was purely on autism research as well, that a lot of studies have fewer than 20 participants. Your study design is obviously very important, and I think this is something we've seen in the previous talks. There's a lot of different pulse sequences out there. You can scan at different field strengths, and I think that if you're comparing to other studies or if you're designing your study, keep in mind what pulse sequence you're using, what the benefits are, what the drawbacks are, whether you should be scanning at 7 Tesla or not. In addition, a lot of studies suffer from small voxel sizes and low SNR, and I think that is a big concern in spectroscopy literature in general, that they have low signal-to-noise ratios, and so the reliability and reproducibility of these measures is limited. There's a lot of variability in the regions studied, and as we've seen, there are different results in different regions. The other thing is the nomenclature. I've seen studies where they use a voxel MPFC, and then you see the same study where they have the same screenshot, and they call it ACC. And so there's not very much consistency in what regions are studied and how they're named, and I think that is important. And the other thing that is a limitation of working with clinical populations is there's variability in the cohorts that you study, whether you have what inclusion and exclusion criteria you have, what comorbidities you might take and might not take, whether people are on medication, etc. The final thing I want to talk about is I would have loved to talk about other edited metabolites, particularly glutathione is an increased subject of interest. We can edit that very well. It is a marker of oxidative stress, which plays an important role in many disorders and can lead to damage of neurons, and so I think that is important. And particularly for looking at functional measurement, NAG as a neuromodulator might be a topic of interest as well. So, to summarize, GABA levels are altered depending upon the disorder, and they're related to psychiatric symptoms, behavior, and cognition, which is an important marker of function and could be a potential marker of treatment. This is probably more relevant than looking at differences in means between groups, but it is important to choose your outcome measures carefully in terms of that they're relevant to GABA and relevant to the disorder. Important to consider when you're interpreting your findings is where does the GABA difference come from? Is it in terms of synthesis or metabolism, neurotransmission, or is it just due to differences in tissue and interneural pathology? And finally, it is very important to think about your study design, your sample sizes, medication, heterogeneity, and comorbidities, as well as the acquisition that you're acquiring and the scan quality. They should be taken into account, and we're sometimes limited by the resources that we have, but these should also always be reported. So with that, thank you very much, and if there's any question, feel free to drop me an email, and I'll stick around as well. Thank you.
That's fantastic. Thank you very much, Nick. We have about 5 minutes if anyone has any general questions Or if people would like to escape, that's also fine. The speakers will be around for a little while, so do come and talk to us. Anybody? Okay.
There's 2 questions from the audience still remaining.
Oh, yeah. Okay.
So this one is for Mohammed. Do you typically see a good correlation between edited measures of glutathione and non-edited measures of glutathione, for instance, steam and press?
Answer is I don't know. So depends. So there's a study done on GABA editing, and they compared unedited sequence with edited sequence. To my understanding, what the paper reported was that you need to have certain signal-to-noise ratio, certain line width, so your shimming has to be good, your quality of the data has to be good. This is, I'm talking about the unedited part, so that it gives you a reliable quantification of metabolites such as GABA. Now, with comparison with edited sequences, again, you need good line width, you need good signal-to-noise ratio data, but it seems to be more reliable because you're editing it. You're actually seeing what you're measuring. Now, when it comes to glutathione, one article mentioned that they reported the results from phantom measurements, and they use a particular software that's Quest, I think, as memory serves, and they found that at higher concentration, press does detect the same level as editing, but lower concentration, then it starts going off measurements. But it depends. Different studies have done different sort of correlation studies. What I would say is that before you start conducting study, do some pilot studies. If you're going to use unedited measurements, compare both and see if you're actually getting measurements that reliable measurements, and that's I would start with that. Yeah. Pilot studies. Yeah.
Oh yes, and I have a question, which is what sequence do we use for the interleaved MRS acquisition? And I'm probably not allowed to just say I can't remember. We had a lot of issues with it, and it's the stories of Polly's PhD, which I'm sure she'll relate at more length than I'm about to. In the end, we did a block interleaved, and we randomized the order between the two voxels across the groups. We did one voxel and then the other, and repeated that. And we used our standard semi-laser acquisition at 70. But the genuine interleaved acquisitions are really, really cool if you can get them to work.
Any question at the back?
Has anyone looked at whether the paired pulse TMS measures of inhibition line up with the MRS measures?
Yes, we have. Yes. So we did a study in 2011 which showed that there was no relationship between spectroscopy measures of GABA and either GABA A or GABA B activity, but that there was a relationship between spectroscopy and a potential marker of synaptic GABAergic tone. Other people have replicated the lack of finding with synaptic activity. We probably can't see the vesicles because of the macromolecules, probably, but no one's reliably looked at the positive finding from that study.
And I believe Ian Greenhouse looked at it and found a relationship with the size of the MEPs.
Yes, yeah, so there's definite relationships that tell us something, but it's definitely not GABA A synapses. I think beyond that, we're all quite interested as to what it might be.
And I think that's one of the things that is a take home, is that with the TMS, you're looking at the GABA receptor side, and we're looking at the neurotransmission and the metabolism side. We know they're related, but to what extent, we don't know. And I think that's an important thing to keep in mind. They're not the same thing. If they're the same thing, you wouldn't have to do both anyway. So.
Yeah, be a lot cheaper. Okay, great. In which case, I think we've probably reached a natural conclusion. Thank you all very, very much for coming and asking questions and getting involved. Thank you for bearing with us through the technical hitches. It's been a great afternoon. I've massively enjoyed it, and mostly fantastic speakers and made it wonderful, but also your input. So thank you all very, very much, and enjoy the rest of OHBM. Most of the speakers are mostly fantastic. Never mind. Cool. Thank you.
Thank you. This was fun.
Fab.
I also learned a lot.
Yeah, no,
Actually, I learned a lot from this talk.
Yeah, no, yes, so bits that I've never presented. No, it's great. Hi.
So I believe in the shamanic intervention,